We are working on the diffusion through the cell membrane state lab. This video demonstrates the first part of the lab, which shows diffusion. In this first part, we will build a model of a cell and observe the type of molecules that move in and out of the model cell. Let's review diffusion. Diffusion is a movement of molecules from areas of high concentration to areas of low concentration without the use of ATP energy. The movement continues until the molecules are equally distributed, known as dynamic equilibrium. Living cells are surrounded by a membrane that is selectively permeable, meaning the membrane allows some molecules to enter and exit the cell while blocking others. This movement depends on the size of the molecules entering and leaving the cell, as well as the internal and external environment of the cell based on concentration gradients. The model cell will be made of dialysis tubing, which will act as the membrane. Remember that small molecules such as water will be able to pass through easily, while larger molecules will not. We are using two indicator solutions, Lugol's iodine and the Benedict solution, to show what molecules are present inside and outside of the cell. We begin with large beaker of water, a glucose solution, starch solution, a cup of water, and a 6-inch strip of the dialysis tubing. First, briefly soak the dialysis tubing in the cup of water to open the ends. Tie off one open end and dip it again if needed to open the other end. Place a full pipette of the glucose solution into the tubing. Followed by a full pipette of the starch solution. Tie off this open end and mix the solutions. Dip the model cell into the cup of water to clean the outside of the cell from any residue. The model cell is complete and can be placed into the large beaker of water. Be sure that the model cell is completely submerged. Using a pipette, add the Lugol's iodine to the beaker of water. Observe what happens to the cell. Lugol's iodine is a starch indicator solution. It is amber in color and turns to a blue-black if starch is present. After 8 minutes, we observe that the liquid in the beaker is still amber, but the cell has changed color from the colorless to the blue-black. Use a pipette to transfer some of the water from the beaker into the two empty test tubes. We are going to perform some tests using our indicator solutions to see the results. In our test tube rack, there are a total of 8 test tubes. The first 2 contain water, the next 2 contain starch, the next 2 contain glucose, and the 2 on the bottom contain the solution from our beaker. Use Lugol's iodine solution and Benedict solution to test the samples in our test tubes. Lugol's iodine solution is a starch indicator, and it turns from amber to blue-black in the presence of starch. Benedict's solution is a glucose indicator, and it turns from light blue to orange only when it is heated and glucose is present. The 4 test tubes with the Benedict solution will be placed in the hot water bath in the beaker. Add the Benedict solution to the remaining 4 test tubes and place them into the hot water bath. After a few minutes, we observed that the first test tube with water remained blue, the second test tube with starch remained blue, the third test tube that had glucose turned a bright orange color, the last test tube with the beaker solution turned a dark orange. The fourth test tube is darker because the solution from the beaker was already amber. After adding the iodine to the test tubes with the 4 different liquids, we observed that the first test tube with water turned amber, the second test tube with starch immediately changed color to blue-black, the third test tube with glucose turned amber, and the last test tube turned to a more pronounced amber. Let's take a closer look at the individual test tubes that had a color change. The first one had starch in it since it turned blue and black, the same color as the inside of the model cell. The second test tube is a bright orange that had the glucose solution. The last test tube is a dark orange because it already had the amber iodine solution. This indicates that the glucose from the cell moved out of the cell and into the beaker, but the starch did not exit the cell. Glucose is a smaller molecule than starch. Therefore, it can move in and out of the cell easily. Iodine is also a smaller molecule, that's why it was able to enter the cell. Hence the blue-black color inside the cell.